Hello there! This video is all about internal validity of research methods, or in other words, being confident that the observed relationship or differences between your independent and dependent variable are reliable and you can have confidence in them. So I will quickly revisit the measures of internal validity and measurement that I've covered in a previous video. Then we will look at internal validity in research strategies and confounds and how some of the threats to internal validity can be prevented. So in the reliability and validity and construct measurement video, I introduced the concept of validity as it applies to measurement or operationalization of variables. The validity of a measurement procedure refers to whether the procedure actually measures the variable that it claims to measure. So there's face validity, which is the participant perceptions of the relevancy or fairness of the items. Concurrent validity, so the relationship between a new and established measure of the same construct. Predictive validity, or criterion validity, which is the relationship between a construct and a desired behavioral outcome, which is often some form of performance. Content validity, where survey items fully capture the construct and measure all of the important elements of whatever attitude, perception, behavior, or experience that you're trying to measure. Construct validity, which is that the measure is consistently related or not related to other variables in ways that are expected and consistent with past research. And then measure sensitivity, which is the difficulty of the scale and the ability to differentiate respondents by capturing the full continuum of the variable. So being sure that the measure is adequate and the sample doesn't have a restricted range where they only represent one end of the spectrum for the variable of interest. So the rest of this video approaches the concept of validity as it applies to an entire research study, the whole strategy, to examine the quality of the research process and the accuracy of the results. So be careful to distinguish between the validity of a research study and the validity of variable measurement. A valid measure in a bogus study is still not going to lead to trustworthy results. So when it comes to the overall research strategy, internal validity is all about making sure that you have one explanation for results. And that explanation is the independent variable, right? So you want to make sure that the independent variable is the reason that you saw differences or changes in the dependent variable. You really want to prevent an ambiguous explanation for the differences in the dependent variable between conditions based on the independent variable or the relationship between those two variables that you're looking at. So this requires that the researcher exert control over extraneous, right? Any extraneous variables, which are basically any variables that are related to the independent variable and dependent variable, but aren't captured in the study. So you can avoid confounds through experimental manipulation, randomization to conditions, and holding all other variables besides the manipulation of the independent variable constant across the conditions in your study if you're doing an experiment. Or in correlational or non-experimental research, you could just measure and monitor the impact of confounds on results. So you really want to prevent those extraneous variables from becoming a confound so that there's no alternative, non-IV based explanation for the changes, the differences in your DV or your dependent variable. Now let's take a look at all the threats to internal validity that are common in behavioral sciences research and think about how to stop them. So unlike a confound, which is typically an aspect of the study, an unmeasured third variable is a variable that's related to your independent variable and dependent variable that tends to artificially inflate that relationship. It makes it look like your independent variable and dependent variable have a stronger relationship than they really do because both are related to something else. So there's a couple of ways to get around this unmeasured third variable. One is manipulate the independent variable, right? That way, you know, especially if you do random assignment to conditions, you're kind of controlling for those variations in things that are related to your independent variable. So manipulating the independent variable is nice because it doesn't rely on just naturally observing the relationship between variables, which can be related to a lot of other things. Another great way to control for an unmeasured third variable in more correlational or non-experimental research is to measure and statistically control for other variables that you think may influence the relationship between your independent variable and your dependent variable. And of course, you're going to need to do an extensive literature review to see what other variables tend to be related with what you're interested in, but it's worth it. So here's a classic example of the third variable problem. If you did some data collection, you would find that over time, ice cream sales are highly correlated with crime rate. In fact, you see here there's a correlation of 0.85 between those two, meaning that on days where there's more ice cream sales, there's a lot higher crime rate. And on days where there's, fewer ice cream sales, there's a lot lower crime rate. But there's a problem with this, right? Both ice cream sales and crime rate are related to a third variable, temperature, right? And if you think about it, ice cream sales are going to be higher on hotter days, and crime rate's probably going to be higher on hotter days because more people are outside doing things. Especially here where I live right now in Ohio, it's cold in the winter. Everything kind of shuts down. We all kind of keep to ourselves, and then it gets real lively in the summertime. So temperature would be the third variable that explains that relationship. Now, if you look at that fancy RXY.Z, that's called a partial correlation. And that's an analysis that you can do where you pull out that third variable to see what the real relationship is between your independent and dependent variable after controlling for that third variable. So I ran an analysis with this data, and as you see here, after you control for temperature, there's basically no correlation between ice cream sales and crime rate, and it actually goes negative, right? So it goes from strong and positive to weak and negative. A more realistic example is looking at the relationship between bottled water consumption and child health. So let's say that you saw a really strong positive correlation, 0.75, between how much bottled water children were consuming, like what proportion of their water supplies from bottled water versus tap water or hose water. That's what I drank when I was a kid. And then how that's related to the health of the child. And that can be operationalized as like the number of visits to the doctor required if you're sick or the number of illnesses or something like that. Let's say we see a correlation of 0.75 there. Well, there's a problem. It may not be that the bottled water is helping kids be healthier, right? Remember, correlation is definitely not causation. But maybe children who come from families with a higher income or a higher socioeconomic status can afford more bottled water. And that money also allows them to afford better childcare, better health care, better food, and just better access to resources in general, which is probably what's leading to the improvements in child health. And in this example, you could see that once you control for income, that relationship between bottled water consumption and child health becomes weak and negative instead of strong and positive. So this isn't experimental research examples, right? These aren't, but you can always measure for the other variables and control for them to really, really dig into what the true relationship is between your variables of interest. So participant confounds are more relevant to experimental and quasi-experimental research studies, but especially to quasi-experiments, because remember, one of the types of quasi-experiments is relying on pre-existing groups. And chances are, if those groups are already within a department or a class or an organization or some other group that already exists, they're probably going to be somewhat different than other groups of people, right? They may have come together because they have similar, characteristics or attitudes or perceptions or behaviors. So it's problematic if you give a manipulation of the independent variable to one pre-existing group and then a different manipulation to the other, and then you assume that any difference you see between the groups is because of the independent variable. Well, probably not, because they were already in groups that were different to begin with, right? So that's a participant confound. An example would be if you were looking at the quasi-experimental example I gave in a previous video, where you went in the break room and you gave the people who were already in the break room some coffee, and then you compared their productivity to the people who were in the break room. Well, chances are that pre-selected group of people in the break room are probably not working as much because they're not, focused on work, and they may already drink coffee, right? So they may be already different from the people that are not in the break room. This type of thing, this participant confound, can also happen with experimental research. And remember, you rely on random assignment, ideally, and you hope that by randomly assigning your people to conditions, that those individual participant differences are kind of evenly distributed. But there's no guarantee. So that's why systematic random assignment can be an advantage when it comes to this, is just making sure that every person in the condition is matched with somebody else in the condition, so all the conditions kind of look the same. So another example would be if you were having one condition, rate their anxiety after watching Saw, a scary movie, and another condition rating their anxiety after they watched Step Brothers, which is a lighthearted, funny movie. You may, before the study even takes place, have people in the Saw group who are more anxious. So let's say like the black shadowy figures represent more anxious people, right? Maybe you already had more anxious people to begin with. Of course, you could measure that beforehand, right? You could do a pre-test, post-test with different groups, and that would help kind of weed that out, but that's something you'd have to think about in advance. Now let's think about how participants can intentionally alter their behavior or responses to have their desired effect on the actual results of studies. So this is all about the reactivity to perceived demand characteristics, and this is also a participant confound. So demand characteristics within the study can reveal or seem to reveal the purpose of the study or the expectations of the researcher. So for instance, if a researcher is watching employees make sales calls, they will be more friendly and pushy just because they know they're being observed, not necessarily due to any interventions designed to increase sales performance, right? Just because they know they're being watched, they're going to try to behave in a different way. So in this study, a sample may react by taking on the role of the good subject. So they may kind of know what the researcher wants to find or have an idea of what their hypothesis is and be overly cooperative and want to help the researcher get their desired results. This is common in volunteers because people who are volunteering to take place in research already have your back, right? And they may want to have your back even more by trying to help you get the results that you want, even though that's not necessarily a good thing. You also have a negativistic subject that could come along, and they're overly combative, and they want to produce data that refutes the researcher's hypothesis. So maybe they're hip to what you're studying, and they want to mess up your data. This is common with coerced participation or participation that involves like anonymous responses and an incentive. They may just quickly go through your survey to get the extra credit. That's why there's kind of some safeguards in place with surveys to make sure no one's doing that, like reverse coding. You could also have an apprehensive subject who's just really overly concerned with appearances and social desirability and tries to portray themselves in a socially desirable way or in a way that they think is acceptable, and they aren't honest or behaving the way that they normally would. So some ways to get around reactivity problems is to use deception in debriefing. So you mislead participants a bit at the beginning so they don't really know what your hypothesis is. And then after the study's over, you can let them know how they were misled and what the true purpose of the research was. But again, after they're done participating. You can encourage the faithful subject role, which is the ideal role a subject plays, by emphasizing the importance of your study for advancing scientific discovery and just strongly encouraging honesty. Again, anonymity kind of helps with that, especially in survey research. You can also try to use a comfortable setting or a comfortable lab setting or a concealed field setting for observations. So instead of watching sales calls, maybe you could record them and not be like in their face and they know that you're there. So environmental confounds are all about the environment that the people are in, right? So you want to make sure that all of the different conditions in your study are participating in the research in the same environment, especially in experimental research. If you're doing a survey and it's online, you can't really control where people are, so that's not as relevant, but you can maybe ask that everybody take the survey in like a calm, peaceful environment, right? To try to stave off those environmental differences that could become a confound. So these differences in the research environment could be due to the actual physical location that you're at. It could be the time of day. It could be even like the temperature, the noise level, the smell, the room size, and even the experimenter, but that's kind of a separate thing when we look at it, the experimenter bias. Just remember, when you're doing research, especially experimental research or quasi-experimental research, try to have everybody participating in the same type of location. And if you're reading a journal article about research, see if they dig into how they held the environment constant across conditions. Experimenter bias is when the researcher unintentionally influences results with inconsistent treatment of sample members due to their expectations, beliefs, bias, errors, or even changes in mood, right? You can see our two different versions of our researcher down here. One's being goofy and one's being very surly and serious. And maybe he was just in a better mood the day that he interacted with condition 2. So experimenter bias really does distort valid measurements, and it reduces the reliability of measurements over time or between conditions because the researcher's treatment of participants influences their responses and is not really measured or controlled for because the difference in treatment is unintentional. So some things to do about it: use scripts so that the researcher is saying the same exact thing in each trial of the experiment, or that, if you have the same form in a survey, everybody's seeing the same thing. Replace a live researcher with video instructions. So maybe just have everybody receiving video instructions from the same researcher, and in different conditions just have slight variations. That would allow you to be sure that everybody in the same condition gets the same experience, and then allow you to maybe even assess any nuanced differences in the video across conditions. The gold standard for reducing experimenter bias is using double-blind research. And this is where the researcher who interacts with participants and collects data directly is unaware which condition the participants are in. So for example, one researcher gives the pills to the participants, and they record who gets a sugar pill, that's the control group, and then who gets some experimental pain medication in the intervention group or intervention condition. And then the participants are unaware which pill they received. And then another researcher comes in who is also unaware which medication participants received. And that's who interacts with the participants to see how long maybe they can endure discomfort, right? So they're measuring pain and their response to pain medication, so they're measuring discomfort. So maybe they have them like see how long they can handle pin pricks or how extreme of a pin prick they can handle, and maybe have them do a strenuous activity and see how long they can handle it. If the researcher who interacts with participants knows what medication people received, they may go easy on the control or the sugar pill condition, knowing that they aren't getting any real pain relief, right? So double-blind research is nice because the researcher doesn't really know which group is which, and so their expectations can't influence the results. Time confounds are sort of related to participant confounds because it's all about how time influences participants. So it's really about looking at how repeated measures or practice or fatigue or even just events that happened between measurements influence results. So for example, let's say that you give a GRE exam, you're doing a quasi-experiment, pre-test, post-test, and on Monday you have students take the GRE. On Wednesday, you give them a GRE review, and on Friday you have them take the GRE again. Well, if you see differences, you could think that, oh, okay, well, they did better the second time on the GRE because of the review, right? But there could have been stuff that happened during the week that maybe didn't make them do as well, or maybe even did help them do better, right? Maybe just taking the pre-test kind of helped with the post-test as well. And, you know, maybe they were in a different mood, right? Maybe there were some participant variables that got in the way. And on Monday, they were feeling pretty good, or they weren't feeling very good. It was the beginning of the week. And then by the time they took the post-test on Friday, they were in a better mood. They were more well-rested. So maybe it wasn't the GRE review that had an influence. It was just changes over time, right? So thinking about that, let's look at the next example where let's say that we have a hypothesis that scary movies are going to result in higher blood pressure than funny or kind of melancholy movies. So we have people come into the lab and we take their blood pressure, and then we have them watch an excerpt from Saw, and then we take their blood pressure again, and then we have them watch an excerpt of Eternal Sunshine of the Spotless Mind, which is very deep and kind of sad, and we take their blood pressure again, and then we have them watch Step Brothers, which is funny and lighthearted, and take their blood pressure again. Well, if we see their blood pressure decreasing over time, and we see that happen, it could be that the movies were having an effect. But it could also be the effect of kind of white coat syndrome, where some people get a little bit tense and have higher blood pressure when they're around somebody who's taking their blood pressure. And maybe by the fourth time they had their blood pressure taken, that experience wore off. And it was just something that went away with time and not necessarily due to the interventions. So that's time confounds. So in summary, internal validity and measurement is all about making sure you measure what you think you're measuring accurately. And then internal validity in terms of the entire study is just making sure that you can be confident that whatever differences or relationships you observed in your data are due to what you think they are, right? Any changes or differences in the dependent variable are conclusively due to the independent variable because you prevented all of these other third variables and confounds. And remember, confounds are going to be due to participants, environment, and time. And then that third variable is more like an unmeasured variable that interferes with the results of your study.